well, who'll be speaking on the subject of reproducibility. Next slide, please. I want to tell you all about John, but I need the next slide to do that. There we go. John is a professor of medicine, health research and policy, and biomedical data science at Stanford. He came to international fame, I would say, many years ago when he published his provocative paper on why the results of most biomedical studies are false, and has continued to spend the rest of his career helping people understand why science is not always as reproducible as we might think. He also, while at Stanford, directs the PhD program in epidemiology and clinical research. He's co-director of a major new initiative at Stanford called the Meta Research Innovation Center, which is looking precisely at areas of scientific reproducibility, has an endowed chair in the Center for Disease Prevention at Stanford University, and is someone who at Stanford is ubiquitous and someone whom everyone respects. I'm really looking forward to hearing what he has to say this morning. John, it's up to you.
Thank you, Mark. It's a great pleasure to participate in this series and to try to discuss the pretty convoluted topic of reproducibility. There's a lot of confusing terminology to start with. People are talking about reproducibility, but they're also talking about replication. And then you can hear exact replication and conceptual replication, reanalysis, repeatability, corroboration, triangulation. All these terms have been out there for a long time, but they're currently being revisited as we're facing new challenges and new dilemmas with new types of studies, new types of data, and new challenges on what reproducibility means. This is showing how often the term reproducibility is used across all domains of science. There's an exponential growth over time in the popularity of this terminology. Basically, we can think of three types of reproducibility. Reproducibility of methods, reproducibility of results, and reproducibility of inferences. Reproducibility of methods is the ability to understand or repeat as exactly as possible the experimental and computational procedures. So if you have some software and some script, if you use the same software and the same script, you should be able to get the same conclusion if you also have the same data. So it's an issue of performing a reanalysis or kind of repeating and getting the same result out of the same package. Sometimes it may not be exactly the same result. So for example, if you have a permutation procedure and you run it 2 different times, you may not get exactly the same result. But if the result is substantially different, then you should start to worry about something not being exactly what you thought it should be. Reproducibility of results is something that usually we use the term replication. It means that we're performing a new study, we are collecting new data, new observations, new participants, new whatever, and we want to produce corroborating results in that new experiment, having followed the same observational or experimental method as we did in the original study. So people will talk about exact replication when they try to do exactly the same thing. Obviously, it's never possible to do exactly the same thing. There will always be some deviation from what was done in the first study, minor or major sometimes, and therefore this leads to potential for debate, and this leads to reproducibility of inferences, which is the making of knowledge claims of similar strength from a study replication or from multiple studies or from a body of evidence. Different people see the same results and they reach different conclusions on what these mean because of different backgrounds, different conflicts of interest, different beliefs, different priors or different understanding on what the results mean. There's differences across scientific fields that affect also what we mean by reproducible research. It's not the same in sociology, in physics, in medicine, in bioinformatics. There's a great variance in the degree of determinism that these fields have. The signal to measurement error ratio can be substantially different. The complexity of the designs and the measurement tools can also vary a lot. The closeness of fit that we are seeking between the hypothesis and the experimental design and data may also be different. The statistical and analytical methods that are used to test hypotheses, some of them get entrenched within specific scientific fields. spontaneity of experimental results may vary. Usually it's higher in social science disciplines than in biomedical disciplines, and higher in biomedical disciplines compared to physical sciences. Whether there is even a culture of replication, transparency, and accumulating knowledge. Some fields are very open, and they're sharing software, code, data, protocols as a sine qua non. Others don't share much or anything. Some fields are willing to perform replications, and they feel that unless they do that, it's not going to be believable no matter what they come up with. And other fields see replication as a me too waste of time. The statistical criteria of truth claims can vary substantially. Many fields in biomedicine are stuck with p-values less than 0.05 being enough to claim a discovery or some sort of finding, while in omics, p-values of 10 to the minus 8 are sought pretty much routinely. Some fields are completely abandoning p-values. They're using false discovery rates or Bayes factors, and some fields are in transition using different modalities that sometimes reach different conclusions when it comes to reproducibility of inferences. Finally, there's differences to the purposes to which findings will be put and the consequences of false conclusions. So some scientific findings may be interested in some very fancy mechanistic view of life or some phenomena, and this may have very little repercussion for life and death, but other studies may make inputs into recommendations that affect the treatment of millions of people and obviously their outcomes, including life and death. This is a map of 20 million papers published in the last 15 years. It also includes 2 million patents, and it's an effort to see how many solar systems exist in this galaxy of science. There's about 200,000 solar systems of different scientific disciplines, and the different colors highlight the 13 big clusters. So life sciences is one such, and physics is another. But if you look closely, there's 200,000 disciplines, and each one of them may have its own rules of engagement and its own understanding about what reproducibility means and how exactly it should be implemented. So when you try to cross the boundaries, sometimes you see that fields that are seemingly doing almost the same thing, they have very different standards about their reproducibility practices. The common denominator across almost all fields is that everybody is trying to come up with significant results. So this is an evaluation of the entire PubMed, worth of 15 million papers since 1990 that we performed and published last year in JAMA. And we found that 96% of papers that use p-values in their abstracts, they claim significant results, statistically significant results. Looking at about 1 million full-text papers in PMC, full-text, again, 96% of them claim significant results, and the vast majority of them say that they're novel. So if you take that literally, scientific discovery has become a boring nuisance. There's just so much of it that we don't really know what to make of it. So reproducibility is probably our best chance at the moment to filter out how many of these millions of claims are real, true, and potentially translatable to something that could help people eventually. Now, in the current environment, this may not be easy because the typical recipe of research practices in most biomedical fields is that you have the prototype of a principal investigator who is solo and siloed. Unavoidably, this means that he or she will be using mostly studies of small sample size, will be cherry-picking one or the best hypothesis to try to get to significant results because this is the way to then seek funding. There's a lot of post-hoc analysis being done. A p-value of 0.05 or slightly less is enough. As I said before, there's no registration of the ideas or of the protocols, very little data sharing, and no replication because replication only has a chance of refuting or not replicating something that is already statistically significant. So people think, why should I waste my time to kill all the work that I did and then not be able to publish it in a high-profile journal? So this is a setting of incentives that don't really align with reproducible science. And this is what you get when you have a field performing in that fashion. This is looking at about 1,800 data sets of fMRI studies, and I have plotted for you the number of foci that have been discovered as a function of the sample size. The only way to discover a lot of foci for different phenotypes or features or tasks is to perform very small studies. When we perform larger studies, these foci go away, where the effects become much smaller, most of those disappear, and it's very likely that most of these claimed discoveries in very small studies don't really mean much. This is not just happening in fMRI research. A couple of years ago, we looked across very different fields across neuroscience, ranging from early basic research, preclinical research, clinical trials, and the common denominator was power failure. We had a paradigm of having very small sample size studies that you expect to have major problems with their reproducibility. This is another exercise looking at neuroscience, psychology, and medical psychology and psychiatry literature, where we did some in-depth text mining of these studies. And the common denominator, even though there is quite some variability across different subfields and journals, is that they have very small sample sizes, which are likely to lead to inflated effect sizes and very often false positive effect sizes, which you need some replication effort to try to see whether they stand the test of replication. This is a completely different field. This is economics. Along with Tom Stanley and Christo Kouliagos, we've looked at every single meta-analysis that we could identify in economics. This is mostly observational economics as opposed to experimental economics. We found 130 topics, more than 10,000 studies, and more than 70,000 effect estimates. The common denominator is, again, very small, very limited power for the majority of these studies and these topics. So you expect to see a similar performance of observational economics and observational neuroscience, even though these two disciplines are so far apart. There's some fields where we do have the benefit of having performed replications in very large scale. And one such field is genetic epidemiology, because in genetic epidemiology, the cost of performing replications has been very low. It's very easy and very cheap to genotype very large sample sizes. And this is in contrast to what is happening in many other fields where running the experiments continues to be very expensive. So in genetics, we could go back when we moved into the genome-wide association era and test what we had discovered in the Candida gene era when we were performing research with small studies, one gene at a time, one hypothesis at a time, or a few hypotheses at a time. The replication rate was 1.2%. So of all these several tens of thousands of papers that had been published in the Candida gene era about associations of single genes tested against single phenotypes, only 1.2% were validated when they were tested with very large sample sizes with very accurate and large-scale genome-wide association studies. Preclinical research has also seen a flurry of results that are not looking very nice in terms of reproducibility. A series of investigations, mostly run by the industry, have shown reproducibility rates ranging from 0% to 25%. The reason that the industry has led these efforts is that they do want to use preclinical research to try to develop new drug targets. So they use these leads from papers that have been published by the best investigators in academia, in the best centers, the best research institutes, and they want to make sense of them and translate them into new drugs or new diagnostics, and it doesn't work. So when they did that in large scale, 0 to 25% of these high-profile papers could be reproduced. So Glenn Begley wrote in Nature in 2012 that the failure to win the war on cancer has been blamed on many factors, but recently a new culprit has emerged. Too many basic scientific discoveries are wrong, and that was his conclusion when his team was able to reproduce only 6 of 53 landmark studies for Amgen oncology drug target projects. Now, what does it mean to replicate or to reproduce? This can be contested, obviously, and we don't really know on whether we have the right answer when we have an original study or when we have a replication, reproduction effort that disagrees in one way or another versus the original study. It's possible that the original study may be correct and the reproducibility effort may be wrong. It could be that both are correct, but they were not really exact replicas of each other, or it could be that both of them are actually wrong and there's some common biases that they had. So regardless of whether they agree or they disagree, they could still be wrong. But at a minimum, when we have large numbers of studies, we can start talking about patterns of reproducibility across different fields, even though single cases will be heavily debated, and this is what I call the reproducibility war. So pretty recently, the first results of the reproducibility project, Cancer Biology, were released, and every single of these reproducibility efforts was heavily contested. So here's one example where the results look very nice. You have an effect in the original study that is significant. You have an effect in the replication effort. The meta-analysis also shows an effect. The effect size is slightly less in the reproducibility, but that's okay. But depending on whether you adjust, for example, for multiplicity of analysis, then the result in the reproducibility may no longer be statistically significant. So one might say that we had powered the reproducibility to be significant, and now it is not. But if you don't correct for multiplicity comparison, which is also fair because this is a reproducibility effort, then it is significant. This led to a huge debate, even though I don't really see much debate. These results seem to be pretty consistent with each other. Here's another example where things are clearly very different. I mean, you see that if you take panel A, the effect size, but also the confidence intervals are very different in the original study and the reproducibility estimate. You have a glass delta of 7.41 versus a minus 0.32, and with pretty tight confidence intervals. Again, this can lead to a lot of debate, and there's lots of things at stake here. It's not just the veracity of the results, but it's also whatever other investment has been made into trying to develop the original study into some translational application, for example, whether new studies have been launched, whether clinical trials are being planned based on these results, whether there's investment, whether entrepreneurs or venture capitalists are thinking about giving money to invest in that original observation that now seems to be so squarely refuted. These concerns have been raised increasingly in a number of fields. Here's a partial list showing reproducibility concerns in preclinical studies on microarray data, on stroke animal studies, general biology, oncology, genome sequencing, mouse in vivo studies, neurological studies, mouse models, genomic cell line analysis, microarray data, commercial antibodies and ELISAs, and the way that journals handle information also. And they pertain to disciplines as different as neuroscience, pharmacology, genomics, bioinformatics, stem cell biology, oncology, in vitro testing, chemistry-led discovery, computational biology, pathology, biomarkers, organizational psychology, and observational research of all sorts. So my field, if I had to pick one out of many where I have failed, is probably epidemiology. And you can see here that 0 out of 52 hypotheses that were claimed in major epidemiological studies were subsequently confirmed in randomized trials. This is a nice paper by Stan Young and Carr a few years ago. This is one of the most famous recent reproducibility attempts. It's the Open Science Collaboration effort where 270 investigators and their teams tried to reproduce 100 experiments published in the top psychology journals. And the results clearly don't look very nice. You can see the comparison of p-values and of effect sizes in the original studies and the replications. About a third of the influential papers could be reproduced. Depending on how exactly you define reproducibility, you may have a little bit of a different answer, but clearly they seem to be different. And even when they could be reproduced, the effect sizes typically were much smaller in the replications compared to the original studies. This has led to a reproducibility war. So there were technical comments and replies in Science a few months after the publication of that very influential study. And some top psychologists felt that there's nothing wrong. This is the way that it should be, and actually the reproducibility effort is wrong, is not what it should be, and there's many reasons, and there were examples being pursued as really not being the best effort to replicate the original findings. However, one would wonder if you have a situation where you have 270 investigators in their teams from the best institutions joining forces, performing something with the best possible methods, with perfect transparency, with everything being reviewed at all stages by the best peers. If this is found or claimed to be so wrong and so suboptimal, what would one think about other studies that really don't have the benefit of that cumulative wisdom and expertise being in them? Here's another example. This is experimental economics. They have also tried to do their reproducibility checks, and their reproducibility results seem a bit better. About a third of the studies could not be reproduced. It's a small sample. It's only 18 papers that have been published in major economics journals, but it seems a bit better compared to what has been documented in preclinical research in biomedicine and in psychology. Now, the difference could be just due to chance, but it could also be due to the fact that experimental economics for a number of years now has followed a number of policies that we think, both based on theory and on some empirical data, that should lead to better reproducibility. So they do have protocols that are communicated. They do share all the software and the code and all the raw data with other scientists, and this is also a prerequisite for publication. So you have a culture where there's openness and transparency, and a lot of thought goes into developing protocols. Plus, you have a culture where you have experimental results where you expect to see better reproducibility compared to just observational data sets, especially observational data sets that sometimes are not even conceived with a particular purpose of looking at one specific question that then gets published. Here's another side of the reproducibility debate. Sometimes effect sizes may not completely nullified upon replication efforts, but they may become much smaller. And this is one classic example where we see this in biomarker studies. If you plot the relative risk in the highly cited study that typically has been one of the first ones or the first one about a biomarker, and the relative risk in the largest study, all the action is below the diagonal with just a couple of exceptions, which means that the largest study finds much smaller effect sizes. They're not null, so they're not relative risks of 1, but they're much smaller compared to what the original study had found. So that biomarker still has some ability to discriminate or predict, but not as much, far less compared to what the original highly cited study would have suggested. Another dimension, as I mentioned before, is reanalysis or repeatability. In reanalysis, we're not performing a new study. We're just going back and re-examining the data from a study that has been done, published, and now everybody can scrutinize because the raw data are available. So there's some experience of repeatability checks, and one of them is an effort that we did jointly with Nature Genetics a number of years ago. Nature Genetics was one of the first journals that made availability of software protocols and raw data a prerequisite for publication for microarray studies. And when we did that exercise, looking at 18 papers that had been published in Nature Genetics, we could not reproduce more than 2 of the 18 papers. Some could be reproduced with discrepancies or with discrepancies from process data or partially, but most could not be reproduced at all. And the reason for the non-reproduction was that the data were not available, the software was homemade and had disappeared, the methods were unclear, so we had several teams of microarray analysts around the world, but they couldn't understand exactly what was being done in that paper, or the result was different, even though everything was available and the methods were very clear and the data were there and the software could be made to work. The result was not what had been published in the original paper. So this is probably disquieting because we're talking about the best field in genetics and one of the best in science at large, and the most transparent field at that time, with avant-garde ideas about sharing of information and software and methods. Here's another field where there has been some experience on reanalysis. Randomized clinical trial data have mostly not been available to other investigators, but this is changing. In the past, it was very difficult to get these data, but currently there's about 20,000 clinical trials worth of data that you can get hold of one way or another. Not necessarily completely free access, but at least you can apply for getting them, and hopefully you can get them. In the past, there were very, very few such trials, and we found 37 reanalyses that had examined the very same question as the original trial in the very same data. And we found that 35% of the time, the reanalysis reached an entirely different conclusion compared to the original paper. So the original paper would have claimed that patients should be treated, and the reanalysis would say, no, they should not be treated, or the type of patients to be treated would be different in the original paper versus the reanalysis. One might think that this might be some enemies of the original investigators who are trying to put them to shame, but actually almost always the original authors were involved also as co-authors in the reanalysis, which means that what we have documented here is likely to be just the tip of the iceberg. Probably many, many more reanalyses of randomized trials were done, possibly in the hundreds or even thousands, but a key attractor for disseminating and publishing the results of a reanalysis has been the ability to say that I found something new. So even though it is the same authors, having found something new and reaching a different conclusion might have been an incentive to publish, which makes things very confusing, but this is the way that the publication system operates, apparently. There's also an opposite movement, moving in the opposite direction of reproducibility and transparency that would facilitate reproducibility checks and reanalysis. And I call this stealth research, which means that some organizations and some entities feel that nothing should be shared, nothing should be communicated, information should be kept as trade secret. There's no need to try to replicate. We will just do things internally. We will make sure that they're fine, and then we will conquer the world. And this is pretty much the example upon which Theranos was built. So when I wrote that paper in JAMA 2.5 years ago, Theranos had published about 100 patents, but practically nothing in the peer-reviewed literature about its disruptive, innovative methods. So I questioned whether this lack of transparency, which does not allow anyone to replicate what they do and reproduce what they do, is the way to move forward. And at that time, they were really very powerful, but within a few months after that, after an investigation by Wall Street Journal and scrutiny by federal regulators and other entities, the company imploded. So stealth research didn't seem to be such a good idea after all. Some potential solutions to improve reproducibility. There's many such solutions, and I have discussed some of them already as I was going through these examples. Some of them we know that they would work very well. Others are very speculative, and we need empirical evidence as to their efficacy. There are some solutions that may sound nice, but they could cost a lot. They could mean that we need to invest a lot of effort, and therefore maybe the trade-offs are not such that we would recommend them. For example, one solution might be to replicate everything. Just keep on replicating every single item that you see produced in science or published in the scientific literature. Probably not a very good investment. That would mean that we would need to either double the research budget or somehow cut our investment in half in terms of what new ideas we can pursue. Or another such idea that I think is highly suspect is try to audit everything that is being done. And this means that I would need to clone myself, and everybody would need to clone themselves so that they have the scientist and the auditor scientist following as their shadow wherever they go in the lab or in their computer to try to reanalyze and reproduce everything that they do. Clearly, we cannot do that. However, there's many things that we could do, and here's a few examples that belong to 12 families of reproducibility efforts that may improve the veracity and the truthfulness and trustworthiness of the results that we get. One option is to promote replication culture at large. So embed replication in the discovery phase. This is what has happened in genetic epidemiology. People don't really separate that much discovery from replication because they understand that replication is part of the discovery. So this is one out of zillions of Manhattan plots that you will see in that literature. In the past, we thought that we had discovered zillions of genes that could affect smoking behavior, and this is what these Manhattan plots are about. In the Candida gene era, there were 359 genes that had been discovered as being associated with smoking behavior. When we performed large-scale studies where we had multiple teams replicating each other's findings and joining all their data instead of just publishing them one piece at a time, we could only validate one of these 359, and we came up with another dozen or so. It's better to have a dozen that we are very certain that they're true rather than have 359 that are completely spurious. If we do replication, it needs to be inexpensive and easy to do. And then who is going to do it? Is it going to be the same investigators? Is it going to be different investigators of the same school? Is it going to be different investigators of competing theories and hypotheses? Do we need combinations of the above? In some easy-to-do problems, maybe we can open replication to the wide public, so lots of mathematics questions, they can be split into tens of thousands of computers, and people could run them in their own desktop or laptop and then accumulate that information at some central facility. These are questions that cannot be answered in a generic term. They need to be seen on a case-by-case basis. I think that there is some general patterns that if you have the same investigators trying to replicate, you unavoidably have some allegiance bias. The same applies if you have investigators of the same school. So one has to ask, how strong is allegiance bias likely to be? Do we have experimental constraints and safeguards that would alleviate the concern for such allegiance bias? And if not, then we probably need to think of different investigators who try to test whether the original experiments were correct and the inferences were valid. Registration is another possibility, and this means that if I do have a hypothesis and a protocol, I would like to register it up front so that people would know that this is what I do, and there would be full transparency about all the steps that I have followed, so that people would compare notes with what was the original intention, and they could also try to reproduce what was done at the first place. This is not always possible. There's a lot of research that I call level 0 research, blue sky science. There can be no registration there because really no one has a preconceived deliverable or hypothesis in mind. People are just testing like crazy very different things, and if something pans out, they will report it. The only thing that I want in that case is some transparency that this is how this emerged, that it was completely unrestricted, exploratory, blue sky science, and there was no plan. This is what I got. Now we need some replication effort, but we can't really claim that there was any specific hypothesis or protocol that was preset. Level 1 would be a registration of a data set, which means that I can share information on how many observational data sets I have available. People would know what the spectrum of multiplicity is in these data sets, so when they see a published analysis about one risk factor with one outcome, they can immediately tell that that was from a data set that includes 5 million variables or a data set that includes 5 variables. That makes a huge difference, because if I have published that analysis with a p-value of 0.049 from a database that includes 5 million variables, then people might easily question whether that p-value is really valid. And unless I have pre-registered that, it's very hard to believe that I just closed my eyes and could not analyze anything else other than that association. Level 2 would be registration of the protocol, and that means that there is a protocol. Level 3 is registration of the analysis plan. and that means that there is an analysis plan. Very often there is a protocol, but there's no statistical analysis plan, and this is decided at a late stage after the data have been collected, and sometimes even after some analysis has been done, which creates some shade of gray into the credibility of the research. But sometimes some of that ambiguity may be unavoidably true, because things don't happen the way that they were intended during the conduct of the study, especially in a study with long-term follow-up and with lots of unpredictable features that may emerge during its conduct. Level 4 is registration of analysis plan and raw data, which means that I also provide the detailed information that I have collected for others to reanalyze, repeat, and also compare notes if they try to reproduce in a replication study. And level 5 is open live streaming, where every day in the morning I just put online what are the experiments and what are the analyses that I'm planning to do for the rest of the day, or I can do that overnight. I can get feedback from the whole scientific community on whether this is a good idea and whether I could improve any of that, and then I revisit the analysis or the experimental plan, and I report the results, and then I put my new set of experiments online, and everybody can comment in a continuing basis. This is one example of a registered report. It's part of the Reproducibility Project Cancer Biology. In a way, these papers have been published before the research was done. It's the detailed protocol of what will be done. Practically, it can also convey what are the tables and the figures that will be in the final paper when the results get accumulated. So everybody can see that this is the original intention, and then can compare notes on whether the original intention was followed, and the paper is populated with the results and published, but there's full correspondence between the registered report before the research is done and the registered report after the research is done. That's some empirical data here on the public availability of research data in high impact factor journals. These are the journals that have the highest impact factor across all science and who publish primary research. And if you see that green color, that's good news. It means that there are policies, editorial policies in place for sharing information and for making also information available upon request, including protocols, and also making this a condition of publication. And lots of color means that there are these policies in place, but the last column shows how many out of 10 of these papers actually adhere to these policies in each one of these journals. And there's lots of zeros there, which means that the policies are in place, but really implementing them is not easy. So very often, many journals say, do make your data available. We encourage you to do that, but actually this does not happen. It's not easy to share data, and many fields have taken the steps to make that easy and common and widespread, but others who have less experience probably see as an obstacle that is very difficult to surmount. And there's always these open questions about who will prepare these data to make them meaningfully available, not just throw them out, and then people would be confused, and probably we would get a lot of confusing reanalysis or reproducibility checks. Then how quickly should the data become available? Is there some sort of enforcement that can be implemented? Should there be some committee or some way to sort out whether everybody or some particular people with particular ideas should be able to access these data? How quickly should the data become available? Within 6 months, within 1 year? Many projects don't really finish, and they can be gold mines to continue to exploit with further analysis. Empirically, sharing does not happen often. This is some data covering the period from 2000 to 2014. We took a random sample of the entire PubMed, and we could see that out of the 500 papers, about half of them included primary data, but of these 250, only one of them had a full protocol that had been published, and this was a separate publication, and none of them had all the raw data available for everyone to use with no restrictions. So even though there's a lot of discussion, and even though there are fields that do have data sharing in place, most of the time this does not happen across the broader biomedical literature. There's many efforts to try to enhance and support and reward some of the practices that are likely to lead to better reproducibility. So one such system is the badge system that has been implemented in the Open Science Framework, and several journals in psychology mostly, but also beyond psychology, have started adopting it. And basically, the papers can get badges for openness, for having a protocol, for having pre-registered protocol, for having raw data available. And you can see here that Psychological Science, a journal that adopted that policy, had a steep increase in the percentage of articles that were reporting available data for other investigators to use. But obviously, we don't know what's happening in other journals, and whether people who do want to have these open and transparent and sharing policies eventually cluster towards the journals that reward these policies and make the other journals even more poor in terms of reproducibility prerequisites. There's a lot of debate about how to enhance reproducibility for computational methods. A few months ago, we published a set of guidelines for reproducibility aspects that involve data, code, and workflows. And the way that we conceive that is a set of different levels of reproducibility. So ideally, you would like to have everything available and also make sure that someone has checked every single step of what is being published. But realistically, this may not be possible, because then you need to have the power and the workforce to be able to go through everything and repeat everything that has been done. Therefore, you can consider different levels, and the lower level would be that someone would at least check that the links where the data are supposed to be, where the software is supposed to be, where the script is supposed to be, these links are functional, and you can get things to move. Now, whether you get exactly the same results, that would take things to a different level that would require more effort and more time and more commitment. Better statistics and methods clearly can improve the reproducibility record. As I mentioned earlier, we can think of improving the transparency of availability of analysis plans. Currently, very few studies have their analysis plans visible before they're done. I think that there is plenty of room to improve statistical training and literacy and numeracy of the scientific workforce at all levels, starting at a very early age. Better study design, some standard features that will promote the trustworthiness of the results. For example, randomization and blinding of investigators in animal experiments are very easy to do, but they're not done. And finally, some checklists that investigators in each field would agree they're totally essential to improve the chances of their research being reproducible. These are data from in vivo studies, mostly on animals, and you see that the vast majority of them don't use blinding for assessing outcomes, and they don't use randomization, even though that would cost nothing and would clearly enhance the chances of the research being reproducible. Institutional practices can also improve reproducibility. Students, postdoc fellows, investigators can be trained on practices that improve the chances of their research being reproducible. And you can see that as some sort of continuous methodological training. Currently, we teach our students and our fellows and investigators mostly about subject matter details, but less so about the core of experimental methods and experimental design and safeguards that would promote the reproducibility of what they do. And also, institutions can have institution-wide policies that would reproducible practices and maybe penalize some practices that are clearly against the needs of reproducibility. Reporting standards can go quite a long way in improving transparency about what has been done. There's about 300 reporting standards that you can find in the EQUATOR website, and each one of them pertains to one type of study design or setting. Many people argue that even though this is helpful, it is a bit late, because once you have a study that has been done, just reporting what was done, and sometimes what was done might have been suboptimal or even foolish, may not be so helpful, and it's also asking people to accept that, oh, what I did was completely foolish. So there's a new wave of such guidance that is focusing on protocols rather than finished studies. And you can also find in EQUATOR a number of such efforts that focus on what do we need to build a good protocol for an X type of design, rather than just wait to report a study that has already been completed. Finally, we need empirical research on how to fix some of these reproducibility problems. So our metric center at Stanford is really trying to put together efforts that are focused around improving research practices and eventually getting more reproducible research. This covers issues of methods, of reporting, of verification or reproducibility per se, of evaluation, and of rewards, because all of these are interconnected in some way or another. And there's many initiatives around the world currently that are focusing on various aspects of that reproducibility puzzle. So there's more appearing almost on a weekly basis, and very often we see that there's some overlap in these initiatives, some redundancy, some not necessarily bad redundancy, because I think that we need more efforts in this sphere, but also some need of coordination of all these efforts that are happening across very different disciplines and across different countries and across different settings. So at the end of the day, maybe to really improve reproducibility, we need to make it have meaning in the reward system. So we need to find ways to re-engineer our reward system, which is focused mostly on productivity. And I think that productivity is very nice. They have nothing against productivity. It's very good for people to be productive and publish and get support for their work and publish more and so forth. But this needs to be tied also with quality, reproducibility, sharing, and translational impact of the research. So I use that acronym of PQRST. It sounds like an electrocardiogram. And currently we're focusing too much on the P wave. Well, we need to find ways to reward these other dimensions that would increase the trustworthiness of what is being produced. Scientists do want to see that done. There's several surveys that suggest that we do care about reproducibility, and the scientific community is not just trying to publish and run away. We do want to get things right eventually. It would be nice to be right at the first place, but sometimes we know that this is not going to happen, and science makes progress through correcting itself. We all do care, though, about getting things right eventually and having reproducible results that we can trust and also apply. I will end with a recent paper that is, I think, a good piece of reading material, the Manifesto for Reproducible Science that we published 3 months ago in Nature Human Behavior. It's a coalition of scientists coming from very different fields, but feeling that there are some common threats and common opportunities around reproducibility in that circle of generating and studying hypotheses, designing studies, conducting studies, collecting data, analyzing data, testing hypotheses, interpreting results, publishing, and then conducting the next experiment and so forth. There are these threats that we can deal with reproducible practices, and these practices would depend on different stakeholders. Sometimes scientists can make a difference. Other times it would be journals that need to take the lead. Funders can be very influential. Institutions can have their share. And obviously, the more stakeholders you have in the game in favor of reproducible research, the more likely it is that you would get the results that you want. All of these stakeholders involved in science have sometimes very different types of interest in research. Some of them want to see results being published, so they want publishable research. Others want fundable research. Some want to see things work, translatable research, and some want to make profit. There's nothing wrong with any of these four ideas about what research should do for us, but we need to coordinate our efforts and to have these stakeholders aligned so that research eventually would be reproducible. To conclude, reproducibility has different meanings across different scientific disciplines. but eventually all of them aim at enhancing our trust in scientific findings. There's many possible interventions that may improve the efficiency of research practices and the reproducibility of the scientific literature. We know that some of them work, but we have very little evidence about many others, so empirical meta-research would be useful not only to assess the prevalence of problems of reproducibility, but also to assess the effectiveness and the potential harms of interventions that try to make research more reproducible. Thank you.
Thank you, John. That was fantastic, and I really appreciate your coming this morning. We already have 1 million questions, well, at least a dozen questions in the question box. Those of you who want to pose questions to John, please type your questions in there. I will try to select questions that I think are going to be easier to answer, and I'll editorialize a little bit in the interest of time, since there's not much time left this morning. First question in the question box says, many of my colleague scientists state that these claims of unreproducibility are overblown. They often say that we know who does poor work and we know who does good work, and in the end, science is self-correcting. So should we not overreact that by shaking the system, we could do much more damage to the scientific process or enterprise, which is so good for society? What is your response? And I guess the corollary question is, how do we know that most meta-research papers are not false?
So I think that meta-research papers have their own share of irreproducibility, no doubt. But I think that reproducibility is at the core of the scientific method, so it's not really something new. I think that science has always made progress because it's focusing on better methods, better scrutiny, better ability to correct itself. So I don't see that as a new message that is disrupting science. In a way, it is just supporting science and offering the real value of science, which is exactly that it can correct itself. There's clearly vast differences across different fields in terms of how much lack of reproducibility we see, and also in terms of what it means not to see a result being reproduced. I think that the connotations are very different, the implications are very different, but at the end of the day, any effort to correct the scientific record more efficiently, more quickly, more rigorously, more robustly is only going to make science look better, not worse. There's some school of thought that says that well, these are difficult times, but it has always been difficult times for science. Unfortunately, science is the best thing that has happened to humans, but this is something that many people don't realize. So let's just forget about this and just try to claim that science knows everything and everything that we publish is correct. Well, this is a very dangerous path, because if we do that, there will be lots of cases that easily some results will be shown to be incorrect. And then we are just giving ammunition to our critics that we were overconfident in results that we shouldn't have been overconfident with. So I do believe that to defend science, we need to defend the scientific method, not some conception of science as being absolutely correct all the time, which is not what science is about.
Okay. One of the listeners says that, in fact, we need to go back to Popper's idea of the scientific method, where we don't try to prove hypotheses, which is impossible to do, but to eliminate alternatives. Any thoughts there?
I think that this is pretty much done already to a good extent, and I think that there's also that higher level of integration of knowledge, where indeed we may be eliminating some hypotheses and making others more likely. I would prefer to see things in a Bayesian view and think of the cumulative evidence kind of affecting our priors and generating some posterior. There are some posteriors that are extremely strong, and I will not be convinced otherwise unless there's amazingly extraordinary evidence to the contrary. So, climate change or tobacco killing people or MMR is safe. I can't think of what evidence would persuade me that these statements are not correct. Many other statements, they just have some modest or high or low credibility, and they have some alternatives that also have modest, low, or or high credibility. And as we accumulate more data, we can fine-tune where our beliefs emerge. And sometimes we need to act based on relatively modest credibility, and we don't need to wait until we know something 100% to be sure, and until we have eliminated every other alternative, because what is at stake may require immediate action, even in the face of uncertainty. But I think if we see things from that perspective, it makes sense that we can have a gradient of certainty and uncertainty for different types of scientific findings, hypotheses, and their alternatives.
John, do you have any thoughts on how we can apply analysis of reproducibility to studies that involve clinical data, I guess particularly EHR data that are collected opportunistically, not as part of a specific protocol, where HIPAA may interfere with our ability to make data public?
I think that there's many opportunities for reproducibility in this sphere. Clearly, EHR data have their own peculiarities, but in essence, there's many data sets, many databases that depend on EHR. And some of them may have incompatibilities against each other, but there's also some crosstalk opportunities. So there are opportunities to compare notes across EHR platforms and data and analysis. And in many situations, there may be opportunities also to compare notes of some of the inferences made from EHR data against other traditional types of designs, including in some cases even randomized controlled trials that may have addressed similar questions. So I think that this is a space where you could think of a pretty robust reproducibility agenda and try to map the performance of these data sets in comparisons within themselves, between themselves, and in comparison with other methods of making inference of these effects.
A listener knows that you talked about reproducibility in the biomedical and behavioral sciences. What can you tell us about physical sciences or chemical sciences?
So there's also work done on reproducibility and related themes in the physical sciences, probably less compared to social sciences and biomedical sciences. But we do have several pieces of evidence. The general perception is that physics and chemistry have higher reproducibility, and to a large extent this is true. There's some simple reasons for that. Usually there's a stronger experimental design, there's more determinism in their data. They have a stronger history of transparency and sharing. So, for example, everything in high energy and particle physics practically is shared with a large community of tens of thousands of physicists who can look at the data, and eventually they all publish their results together. They have very stringent statistical rules for claiming discoveries. So they have a 5 sigma rule that is equivalent to a p-value of 10 to the minus 7, which is very close to the genome-wide level of significance that is used in biomedicine, that has worked pretty well in genetic association studies, for example. So there's many reasons why you see pretty good reproducibility in these sciences. There's also great tolerance for negative results. Most of the results in experimental physics are negative practically, and nobody worries about that. You know, you see all these papers coming out from CERN, hundreds of papers. They have found one Higgs boson and pretty much nothing else. That was a great discovery, but I would say it's an equally great discovery that all the other results are negative and people just say that this is what they are. There's still some problems in specific fields that the experimentation or the observation is more difficult. For example, claims for exoplanets, about 50% of the time, they turn out to be non-reproducible, range from 25% to 60% in different surveys. Some of the more recent wave of chemical synthesis have been a bit difficult to reproduce in independent labs, but I hope that this is the exception. So no scientific field is immune to these problems, because these are not necessarily problems. This is the scientific method, and and it's difficult to do good science.
That's great. And on that note, we should end because we've reached the end of the hour. John, let me thank you for what was a really stimulating talk. And let me thank all of the people who dialed in this morning. It was really great to hear you. Thanks again.
Thank you.